All right, let's jump right in. Today, we're going to dive into how scientists can write custom biological messages, called mRNA. We're talking about going from a simple piece of code on a computer all the way to a working instruction inside a living cell. So how do they do it? Okay, let's start at the finish line. What you're looking at right here are actual human cells, and they're glowing this brilliant green. And you have to ask, why? Well, it's because they've just received an instruction manual that we built in a lab, and they are following those instructions to a T. This technology became famous thanks to the COVID-19 vaccines, but it's so much bigger than that. It is a total game changer. It basically lets us give ourselves a recipe to create almost any protein we can dream up, from medicines that could fix diseases to vaccines that train our immune system. But hold on, before any of that magic can happen in the lab, the message itself has to be designed. And this is really where biology meets computer science. It's a lot like a software engineer writing code for a new app. Scientists have to meticulously design the genetic sequence for the mRNA. So every single mRNA message is built from 5 key parts, kind of like a well-written letter. First, you've got the 5 cap, which is basically the big start here signal for the cell. Then you have these regions called UTRs that act like the introduction and conclusion, controlling the pacing. The coding sequence, that's the main event, the actual instructions for the protein. And finally, you have the poly A tail, which acts like a bodyguard, protecting the message and making sure it sticks around long enough to actually get read. Now, here's where it gets really clever. This digital recipe gets fine-tuned. Scientists run it through software that swaps out rare genetic words for more common ones, which helps the cell read it way faster. They also snip out any sequences that might accidentally trip the cell's internal alarm systems. It's basically like running a spell check and simplifying the language to make it as clear and effective as possible. Okay, so the digital design is locked and loaded. Now it's time to move from the computer to the lab bench. We're about to take that digital blueprint and actually manufacture the physical mRNA message. First things first, we need a physical master copy. That digital design gets turned into a much more stable DNA template. Then, using a technique called PCR, which is pretty much a molecular photocopier, we make millions, even billions of copies of that template. This just ensures we have more than enough blueprint to work with for the next step. And this image, wow, it says it all, doesn't it? On the left, you see a perfect, clean DNA blueprint, just one single sharp band. But on the right, it's a mess. Lots of extra junk in there. It's simple, really. If you start with that messy template on the right, the whole thing is just doomed to fail. Garbage in, garbage out. So, with our perfect blueprint in hand, we bring in the master builder, an enzyme called RNA polymerase. You can think of it like a biological high-speed printer. This amazing little machine grabs our DNA blueprint and just starts printing out thousands of copies of our final mRNA message, building them one genetic letter at a time. Okay, so now we have our precious message. But here's the catch. If you were to just inject this raw mRNA into the body, it would be totally useless. It's a super fragile molecule, and the body's enzymes would shred it in seconds. It would never reach its destination. It needs protection. So what's the solution? How do we package it? Well, scientists came up with this brilliant idea to wrap the mRNA in a tiny protective shield made of fats, or lipids. This little bubble is called a lipid nanoparticle, or LNP for short. And the way they build this is, honestly, it's almost like magic. It's like a self-assembling shipping box. You take the mRNA, which is in a water-based liquid, and the lipids, which are in an alcohol-based liquid. Then you shoot them together at high speed inside a tiny microfluidic chip. And just like that, the lipids spontaneously wrap themselves around the mRNA, forming these perfect protective nanoparticles. It all just happens. So we've designed it, built it, and packaged it. But is it any good? Well, every single part of this process has to pass some incredibly strict quality tests before it gets anywhere near a living cell. And hey, just like we checked our DNA blueprint, we have to check the final mRNA itself. What we want to see is that beautiful single sharp band on the left. That tells us our message is intact and ready to go. The smeared mess on the right, that's degraded mRNA. It's basically a shredded letter, completely unreadable. And the final packaged product gets its very own report card. We check 3 really critical things. First, particle size. Are they the right size to get into cells? Pass. Second, encapsulation. Is more than 90% of our mRNA actually tucked safely inside that bubble? Pass. And third, uniformity. Are all the particles pretty much the same? Pass. If it gets 3 passes, it's ready for the final test. After all of that incredibly meticulous design, manufacturing, packaging, and testing, there is only one question left, and it's the only one that really matters. Does it actually work in a living system? To find out, we deliver our final product to living human cells. Now, this particular mRNA has instructions to build something called green fluorescent protein. At a pretty small dose, you can already see some of the cells got the message. They're starting to glow. It's working. So, what happens when we up the dose a bit? Well, the effect becomes much more dramatic. Way more cells are reading the instructions, and protein production is ramping up big time. The green glow is so much brighter and more widespread. And at the highest dose, well, you can see for yourself, it is a resounding success. Nearly every single cell is glowing bright green. This is the proof. This is what shows that the entire journey, from a digital sequence on a computer screen to a real biological function inside a cell, worked perfectly. You know, we just walked through the whole process for making one single protein, but this platform can be used to write almost any biological message you can think of. So it really brings us to the most exciting question of all. Now that we can give our bodies instructions to make almost anything, what should we tell them to do next? The possibilities, they're really just beginning.